Hey everybody, Barton's Biology, DNA, form, and structure. Here we go. Let's crush it. So, let's start off with a bit of a hypothetical. Let's imagine that you are an adult and thinking about having a child. What if for $150 you could take a test that would tell you whether your child would excel at sports, have a high IQ, or look a certain way? Would you take that test? Think about that. For $150, maybe excel at sports, maybe a high IQ, or look a certain way, would you take that test? And then just think about why you answered the way that you do. Yes, because this. No, because this. Why did you answer the way that you did? Now, in reality, tests similar to that already do exist, and they are more common and more affordable than you might think. As an example, before my son was born, uh, my wife and I had some genetic testing done to see how compatible our DNA was and the likelihood that our prospective child might have any number of genetic diseases or disorders based on the genes in our DNA. That test was not terribly invasive, um, and covered on insurance, and it was interesting information to see. Now, notice the difference here. This was genetic information to see the likelihood of diseases in our child. Right? Now, that is different, certainly, than seeing things like if your child might excel at sports. But the information is locked away in the same place. It's in our DNA. So, what is DNA? DNA is a double helix. It is kind of like if you imagine a ladder and twist it, right? Just a regular step ladder, twist it. That's what a double helix is. That's what the DNA double helix kind of looks like. Bases facing each other. Deoxyribonucleic acid is what DNA stands for. And it is made of many small parts called nucleotides. DNA is a nucleic acid, and nucleic acids are a fundamental building block of life. It is one of the groups of macromolecules like proteins, lipids, and carbohydrates. There are three parts to a nucleotide: a phosphate, a 5-carbon sugar called deoxyribose, and a nitrogen-containing base. Those four bases, there are four different types of base. It could be any one of these types of bases to make a nucleotide: adenine, guanine, thymine, and cytosine. Now, those four bases make up the genetic code A's, C's, T's, and G's. In the 1940s and 50s, there was a scientist named Chargaff, and he noticed that the amount of adenine was the same as thymine, and guanine was the same as cytosine. So now we have what's called Chargaff's rule. And it just states that adenine and thymine pair together and cytosine and guanine pair together. So anytime you would have a C or a cytosine on one strand of DNA, that would pair with a G on the complementary strand. Here's what those strands in the helix kind of look like. The nucleotide there in the white box. You can see the G is paired with a cytosine. The A's are paired with thymines, and so on. Two bases paired together we call a base pair. Okay, and it's the sugar and phosphate that make up the backbone or the sides of our ladder. So now, a little bit of review. Test yourself. Think about these questions. The D in DNA stands for what? Chargaff's rule states, summarize that. And if this was one strand of DNA, one side of the double helix ladder, what would the complementary strand read? So ATTGCA and so on. What would the complementary strand of DNA read? That's it to get us started on the structure and form of DNA. Later in this unit, we will continue just exactly with more specific information about DNA and how our cells use it. Have an awesome day. I'll see you next time.